Nero Rice. I work at UniProt at TI as a software developer, and I'm going to talk about some new data sources and integrations within UniProt. Some third-party visualizations are being embedded in entry pages, and also some upcoming changes to UniProt KB and to proteomes, reference proteomes. But first, I will first talk about what is UniProt. Just read out the mission statement. So the mission of UniProt is to support biomedical research by providing a rich resource of freely available, stable, comprehensive, and accurately annotated protein sequence and functional information. And to that end, we have a set of databases to support that mission. The first one being UniProt KB, which is our most popular one. And this is split into two parts. And there's the SwissProt, which has the expertly manually created annotations. And then we have Tremble, which consists of computer annotated using ARBA or UniRule. And then we have the proteomes database, which is sets of proteins for a given species. And within that, we have 25,000 reference proteomes and 900,000 non-reference proteomes. And I'll talk a bit about this later on. And then down, then we have UniRef clusters. So this is clusters of sequences from UniProt KB and UPark, and they're clustered by sequence similarity. And there are 750 million clusters within that database. And then we have UniPark, which is a comprehensive and non-redundant database of protein sequences. So it's a giant archive of 1 billion sequences. And talk about some of the tools we have. In the top left, we have BLAST. So you can search your sequence by similarity against UniProt KB, UniPark, and UniRef. And the top right panel, we have Align, where you can align multiple protein or nucleotide sequences using the Cluster Omega program at EBI. And then we have Peptide Search, which is searching UniProt KB for protein entries that exactly match your peptide sequences. And then the bottom right panel, we have ID mapping, which allows you to map identifiers both within UniProt and external to UniProt. And so for all of these tools, if the results are within some sort of UniProt database, you can customize those results, changing the columns. You can do some faceting and perform additional searches to filter on those results. And a new feature we released this year is using a technology called the shared worker. And so with this, if you have multiple tabs in a browser with UniProt open, if you submit a job in one tab, then that will be populated in all the other tabs. You don't need to refresh the page. So when the job is done, it doesn't matter where you are, it'll tell you your job is done. And now I'm going to talk about some new data sources and integrations. So the first one here is homology from the Alliance for Genome Resources. And this is located within the similar protein section of UniProt KB. And so we're actually loading this data directly from AGR, and we load it dynamically so as the page loads. And we're trying to reflect the same information that you can see in the AGR site. So for example, here we have the 12 methods that have predicted orthology for each of the given genes. And so a blank circle means that it's been checked and it's not been predicted, and a solid circle means it has been checked and it has been predicted, and the dash means it hasn't been checked. And likewise for the paralogs tab there. And looking now at the feature viewer tab within within the UniProt KB. So we have some new information coming from the PTM Exchange Consortium. We have new modifications such as phosphorylation, sumylation, ubiquitination, and acetylation. And these are located in two places. The first track there for the PTMs, it will show you the residue that's changed. And then within the second track that's pointing out there is PTM containing peptide. That shows you the residue that's changed within the context of the peptide. So the little line there is the residue that's changed. And then we also have some information. We have a new track from epitopes. So this is coming from the immune epitope database. And this is experimentally characterized adaptive immune epitopes from large-scale studies. And we have a new RNA editing track. And so this is coming from the Ready portal. And this is data derived from large-scale RNA-seq transcriptomic analyses. And it's showing 8i RNA editing events. And it's currently human. That will, I think it's in the pipeline to change in the future to encompass more species. And we have some information from the Human Proteome Project. And so this is allowing you to view peptides that have been experimentally confirmed through mass spec and then mapped onto the human protein sequences. And then lastly, we have some information from some data from Google DeepMind. First of all, we have AlphaFold. And so the track showing here is showing the confidence of the predicted structure prediction. And then below that, we have alpha missense, which is predicted pathogenicity for that particular amino acid. And then also with Google DeepMind, we actually show the predicted structure here. This is also within the sequence series, just a bit below the previous screenshot. And so within UniProt KB, we have community annotations. And we have improved the way that these community annotations are shown. We've merged things together. We had a lot of duplications. We're showing the submission date. And also it's searchable now using this community PubMed ID. And we have some embedded third-party visualization. So this has been, this is Complex Sphere developed by the molecular interaction community. And this shows the different proteins involved in forming a complex. And if you click or expand on a protein, for example, this is GSK3B, it will show you the exact amino acid where the binding happens if it is known. And so we have this for 10,000 UniProt KB entries. And the Go ribbon, we've had this before, but we've had some improvements. So with the Go ribbon, you have a sling set. And this is designed to cluster more detailed Go terms into a broader category. And so it gives you kind of a simplified view. And what we've improved here is the slimming set name is more intuitive, and also the default will be specific to the UniProt KB entry you're at. And we also are having GoCam models. So this is developed by the Gene Ontology Consortium. And this is data obtained from them, and we filter for UniProt specific curated models. And it provides structured representation showing how the molecular functions of gene products are causally interconnected within the biological process. And we have this for 4,000 models for over 2,000 UniProt KB entries. And just a quick view of the means of data access. So we have 4 main ways of getting to our data. The FTP, which is just big data dump, everything in one go. That is resumable, but you can't customize just everything in one go. And then we have the stream API, and this is limited to 10 million items, and you can customize it, but it's not resumable, so you can't stop and start it. And we have paginated, and that can be as many as you want. So it's fetching a page at a time, and this is customizable. I mean, you can change the columns and so forth. And then lastly, we have a relatively new thing, which is API file generation, which has no limit. So if you wanted to search over UniProt KB and you want to have the JSON for the whole thing, you can do a star search and then submit that job, and then our backend will produce it, and it will tell you when it's ready, and you can then download that. And so that's also customizable. And something new this year is we have Swagger documentation, just a screenshot there, and there's the URL. And so this shows you the different ways you can interact with our different endpoints within our REST API. And we have a new XREF. This is CARD, the Comprehensive Antibiotic Resistance Database from McMaster University. So here we have an example, UniProt KB protein CTXM1, and that's linked to this CARD entry. And so CARD shows it is an extended spectrum beta lactamase that confers resistance to the drug class cephalosporin antibiotics through inactivation. So what users can do is they can find all of the antibiotics-related proteins using this query, just database CARD, and you can also be more specific and look for a particular mechanism of action. And coming soon will be a knowledge graph to show resistance context from card. And we're always adding new cross-references. Here's some we've added in the last year. So if you'd like to have your cross-reference with UniProt, please visit this link, which is to our link tree, which has just been set up. And from there, you can contact us. And yeah, love to hear from you. So some upcoming changes. I'm going to talk about reference proteomes and the way that we're improving how this is being computed. And also in the future, UniProt KB will consist mostly of reference proteomes, and how this will help remove noise and improve searchability within UniProt KB. And also, so Tanushree Tunstall is in the audience somewhere. She's worked on this project amongst others. So if you have any questions, do reach out to her. And first, I'm going to have a quick review of how we import proteomes and proteins into UniProt. First of all, it starts from INSDC, which is the International Nucleotide Sequence Database Collaboration, composed of ENA, DDBJ, and GenBank. And so we get a complete genome, and then we get the protein sequences, which are derived from that genome annotation. And then we perform some manual filtering, but the vast majority of, so for example, the Salmonella, which we have a lot of Salmonella sequences in UniProt. So for this example, the majority of these sequences go straight to UniProt because UniProt is an archive, so it's there on purpose to be archived for a long time. And then from here, from UniProt, we have a few checks, checking for redundancy and quality, and then we'll go to UniProt KB. So we go from 9.4 million to 2.5. And then from here, we are annotating either manually expert curation through into SwissProt, or we will use the automatic annotation methods, UniRule and ARBA. And so with these UniProt KB sequences, we group these by genome assembly, and then we form proteomes. And so then we come to the task of trying to determine a reference proteome, which is a proteome that best represents the protein space for each species. And so we do some novelty filtering, and then sequences are clustered by similarity. And then finally, there's some manual curation, and finally, we say this is the reference proteome. And so just going to talk about some challenges with that. First of all, there's the explosion of data from biological sequencing. It's because sequencing is becoming cheaper and faster and everyone is doing it, there's just an explosion of data. And so we have more genomes coming from INSDC. And so that then impacts UniProt KB. And you can see in this plot, the massive spike coming up here. This is the number of entries, and it's growing over time. And so the consequence of that is UniProt KB can be like there's too many needles in the haystack. There's some redundancy, and there can be some noise. And so the challenge for reference proteomes is that the current pipeline can't scale with such an abundance of proteomes and proteins. And another issue is that sometimes there will be 0 reference proteomes available, or there could be many. So for example, for Salmonella, we go from 15,000 proteomes, and we come down to 300 reference proteomes, which is still quite a lot. And so also another aspect that is important here is that the proteome completeness is not considered in the determination of being a reference proteome or not. And so to that end, we have a few changes coming up that we're planning. And the first one is to improve the way that we decide reference proteomes. And it's a new scalable workflow. And for cellular organisms, we will use MM62, which is a many-to-many sequence search and clustering algorithm. So it's super fast. And using this, we can basically use the majority of the UniProt sequences for a given species and basically compare all against all to determine a clustering. And by doing that, we replace some of the more ad hoc filtering we had in the past. And this allows us to select the most complete, high quality and representative proteome per species while maintaining stability. And also at the same time, we're going to ensure there's at least one reference proteome per species. And for viruses, we would just align with the International Committee on Taxonomy of Viruses. So whatever they consider to be a exemplar genome, we will term that to be a reference proteome in UniProt. And so for UniProt KB, we want to make a more focused UniProt KB around high-quality reference proteomes without compromising manually created knowledge. And so we will have UniProt KB will consist of proteins from reference proteomes, SwissProt entries, entries with PDB cross-references, and other proteins of specific biological interest. And for non-reference proteomes, they will, as always, be accessible within UniPark, so they will not be lost. And we plan to do this February 2026, but we started to do some transitioning now. So in the next release, which will happen approximately September, we are ensuring there's at least one reference proteome per species. So to give you an idea of what this will look like, in this view here, we have the tree of life, and each image represents a proteome, and then each emboldened image represents a reference proteome. So in UniProt KB now, you can see that in some situations you have many reference proteomes, you may have none, you may have no reference proteomes. And then coming to the proposed plan of changes, is we will have a more evenly distributed, broader coverage set of reference proteomes. And so the tree of life will have 34% increased number of species with reference proteomes. And so we will have some user interface changes to support this. In UniPark, we will have something we're calling a cross-reference entry page. So this could be for any UniProt KB entry, both reference or non-reference. And this is a plan for the future. This is not available at the moment. And this will show you information that's derivable from the sequence. So for example, you have interpar reference domains. And we're also planning to have on-demand annotation with the unifier. So a user would click, say, please annotate with unifier, which is under the hood is using ARBA and UniRole. And we've done some work already. So we have a more proteome-centric FASTA format available within UniPark. And we also have a UniPark Lite endpoint, which improves performance and scalability and works with many extra cross-references. So right now, we already have quite a few pages with thousands of cross-references and can't even load the page. It's too many. So this new Lite endpoint allows us to paginate those. And so the page loads now. And then something coming soon is to look at the pan proteome and refactoring that. So this is the UniProt team. It's a collaboration with EBI, PIR, and SIB. And here are our funders. And this link up here takes you to a link tree where you can talk to us about the reference proteome changes. And there's other surveys there, and it also has the details of additional UniProt talks coming up. Yeah. Also, yeah, Tanishree's here to answer any questions. So any questions from you guys?
We've probably got time for questions. Yeah. Yeah, so we've got time for one or two questions. We've got about a minute or two.
Yeah, that's a great talk. For the PTM data, so where you get the data from, like PTM side or PTM type, and how do you do control?
So this is coming from PTM Exchange. I don't have the details of that at the moment. I don't know if anybody here does have that information.
Yeah, can you repeat the questions?
Yeah, so like for the PTM data, so where you get that for PTM site and also PTM peptide. So we know there are many kind of type of PTMs.
Yes.
And how do you do quality controls for that data?
Yes, so there is a paper that we can then pass to you of how the PTM exchange is developing the methods to reanalyze proteomics data sets with post-translational modifications. So they run the pipeline with the peptide atlas database, and then we take the best PTMs from those proteomics data sets. But we have a paper about it, so I can just pass that to you because it's complex.
Okay. Yeah, thanks very much, Daniel.